Hello and welcome to this Nature.com custom webcast titled Analysing Ebolavirus glycoprotein and its interactions with therapeutic antibodies using MALS. My name is Jayshan Karpan and I will be your moderator. Today's webcast is sponsored by Wyatt Technology Corporation. Wyatt Technology is the recognised leader in instrumentation for determining the absolute molar mass, size, charge and interactions of macromolecules and nanoparticles in solution. These tools include inline multi-angle static light scattering, high-throughput dynamic light scattering, differential refractometry, electrophoretic mobility, differential viscosity, field flow fractionation, and automated composition gradient. We'll begin today's webcast with presentations from two speakers and end with a Q&A session where we will address the questions that have been submitted by you. To ask a question, just press the Ask a Question button at any point during the webcast and we will answer them today. It is now my pleasure to introduce the first speaker, Dr. Daniel Sohm, Principal Scientist at Wyatt Technology Corporation. Over to Dan.
Thanks and welcome everyone. It's my great pleasure to be here with Dr. Kate Hasty of the Ullman Sapphire Lab at Scripps Research Institute. She'll be presenting to you a little bit later on some really interesting measurements and analyses that she did on the interactions of therapeutic antibodies with the Ebola glycoprotein. And what I will be doing is first giving you an overview of the light scattering technologies that she was using in her measurements. And so, what can you learn with light scattering using the two techniques SEC-MALS and CG-MALS? So in the course of your biomolecular research, if you're producing proteins and other biomolecules, you need to ask certain questions of those samples, and you need to know the structure and solution properties, the molecular weight, the native oligomeric state, if the protein is glycosylated, to what degree. Sometimes you need to know information about its conformation. How does it function? So how does it interact with itself and with other proteins? And finally, if you're planning on using that material for other types of research which may involve heavy investments of labor or perhaps costly beam time on a neutron scattering beamline, you want to make sure that before you go into that additional research that your protein and your biomolecules are suitable quality and that the end results will actually be viable and reliable. So to make sure that your sample is pure, it's not aggregated, that it's stable and will last throughout those additional measurements and of good quality. And light scattering can help us address all of these questions, and you'll see that in the course of the coming slides. So what can light scattering actually measure? We distinguish two flavors of light scattering. The first is called multi-angle light scattering, which is also static light scattering. And it is used to measure from first principles the molecular weight of molecules in solution. And often it can also determine their geometric size in terms of a quantity called the root mean square radius, or RG, radius of gyration. And it does that using the equation shown here, simply by illuminating the solution with a laser beam and measuring the scattered light. We can determine the molecular weight if we know the concentration of the molecule and we know a quantity called dNdC, that's the refractive increment, and that is a universal property of all unadulterated proteins. Once you apply some post-translational modifications, it might require some changes, but for standard proteins that's a universal number, and so simply by measuring concentration and scatter intensity we can determine directly the molecular weight. If there are multiple species in solution, then what we measure with MALS, or multi-angle light scattering, is the weight average molecular weight. So it'll be an average of molecular weights which is weighted towards the more massive proteins. By measuring the angular dependence, that is, how does the intensity vary with angle relative to the direction of the laser beam, we can determine the size, the root mean square radius, if the molecules are larger than about 10 nanometers in radius. Now many of your proteins will be smaller than that, and then that is where dynamic light scattering comes into play. Dynamic light scattering, instead of simply measuring the intensity of the scattered light, actually measures the fluctuations in the scattered light. And those fluctuations arise from Brownian motion of the molecules. They're jittering around in solution due to Brownian motion. And the rate of fluctuations will inform us about the diffusion coefficient. And from that, we can then determine a measure of size called RH, the hydrodynamic radius. And RH can be measured from 0.2 nanometers and up to several microns. So it is certainly appropriate for most proteins and even peptides, even smaller peptides. There are also some derived measurements that we can determine by combining dynamic light scattering, multi-angle light scattering, and other information. So when we apply these techniques to size exclusion chromatography, we can actually characterize each of the components that elutes from the column. And by combining the information on size and molecular weight, we can learn something about the conformation of the molecules. We can also utilize multiple detectors to characterize multi-component complexes. So for a standard species, which consists of, say, a single type of protein or a single type of polymer, simply by measuring the concentration with a single concentration detector and light scattering, we can determine molecular weight. That doesn't really work when you have a conjugated complex, for instance, a glycoprotein that combines sugars with standard protein, or a membrane protein, which is eluting bound to the surfactant that is used to solubilize it, or pegylated protein. In order to characterize these, we need two types of concentration detectors, and typically what we do is use a combination of ultraviolet and refractive index, and together with the light scattering, we can actually determine the molecular weight of each of the components, say of the proteinaceous component and of the modifier. We can also obtain derived measurements in batch mode, which does not involve fractionation. So in batch mode, we're actually going to measure the weighted average molecular weight of the solution as a function of concentration and composition, and from that we can learn about interactions, because as you change the concentration or composition, you'll have changing degrees of formation of complexes. Could be homoassociation, could be hetero-association. By analyzing the change in molecular weight with concentration, we can determine the stoichiometry of the complexes that form, say are they dimers or tetramers, and the KDs, the equilibrium dissociation constants. So these are the things that light scattering can measure for biomolecules in solution. And there are two techniques that are used to perform those measurements. So the first is SEC-MALS, or SEC-MALS as I call it, which combines multi-angle light scattering and actually online dynamic light scattering as well, with the separation from size exclusion to analyze the solution components. So the system comprises a standard HPLC. You can work with most types of HPLCs or even FPLCs, as long as you can put the size exclusion column in line with it. We typically use the UV detector from the HPLC system and add on downstream of that the light scattering detector, a Wyatt Dawn or Mini Dawn, and a refractive index detector, which is a universal concentration detector used for characterizing all types of molecules, and that would be the Wyatt Optilab refractive index detector. Inside the light scattering detector, very simply, we have a laser beam illuminating a solution. So as the eluent is coming off the column and going through that detector, the different molecules come into the laser beam, and we'll have multiple detectors at various angular positions, and they will simultaneously collect the scattered light, determine the intensity, and from there the software, the Astra software, will collect and interpret the data to give you molecular weight, size, and all the other properties that we can determine with SEC-MALS. So why go to all the trouble of adding these extra detectors when you have perfectly good size exclusion chromatography? The problem is that if you're using size exclusion chromatography, you're relying on assumption that the elution of the molecules from the column can be directly related to their molecular weight. And sometimes that works, but often that doesn't work. And the reason is that SEC doesn't actually separate based on molecular weight. It separates based on hydrodynamic volume. And so you're assuming a relationship between hydrodynamic volume and molecular weight, which may not be true for your molecules. And also other things, non-ideal interactions happen with a column that will change the elution time in ways that the reference standards do not interact. So you're making various assumptions in standard column calibration SEC, assuming that the molecules have the same conformation as the reference standards, the same density, the same degrees of hydrophobic or other types of non-ideal interactions with the column. And so all of these are often untrue for your actual samples relative to the standard globular proteins that are used for column calibration. You're also assuming that the chromatography is reproducible day to day. If not, you have to regularly recalibrate your column. And these are all things that are avoided by the use of light scattering, because light scattering really doesn't care about the specific elution time. It's not fooled by non-standard molecular conformations, by column sticking, by variant chromatography. And to the right here, we have a nice example of this. We have the elution of native RNase protein, which comes out relatively late, and the reduced RNase protein, which comes out much earlier. So based on column calibration, you would think that the reduced RNase protein is actually much higher, has a much higher molecular weight than the native protein. But that's not true. It's simply unfolded and changed its hydrodynamic volume, and therefore it's eluting much earlier, even though it has the same molecular weight. The light scattering actually gives you the information directly that these two peaks have the same molecular weight. So it's not fooled by the difference in elution time. So here's an example, another example of where MALS gets it right. This is these are the chromatograms of two proteins, apoferritin and IgM. They have very different conformations, and they have different relationships in terms of elution time relative to molecular weight. And so we can see that the apoferritin has this primary peak, eluting here, and some secondary peaks, which indicate aggregation. The light scattering analysis, that's these points over here, first of all show us that across these individual peaks, the molecular weight is actually uniform. So information that you can't learn from standard SEC is how uniform the peaks are. Here the light scattering tells us that we have very homogeneous solutions in each of these peaks, and so these are monodispersed peaks, and then shows us the molecular weight, which ranges here from the tens of thousands all the way up to aggregates, which are in the tens of millions. In fact, MALS can analyze molecular weights from a couple of hundred Daltons up to hundreds of millions of Daltons, so that's quite a large range. And it can measure sizes from 10 nanometers up to 500 nanometers in terms of radius in it, and that you can also add dynamic light scattering to look at other ranges as well. The IgM, similar, distinct peaks which are uniform, indicating homogeneity across each peak, but you'll notice that the dimer peak of the apoferritin and the monomer peak of the IgM elute at very different times, even though they actually have very similar molecular weights. And that is because of their different conformations. Again, standard elution analysis and column calibration would give you erroneous results, but the light scattering gives you correct results, and you can be confident in your analysis. Okay. And I mentioned that we can analyze protein conjugates as well using the triple detector system. So for glycoproteins and pegylated proteins, other types of modified proteins, we use 3 detectors. To analyze the concentration, we'll use a UV detector, which will typically be sensitive just to the protein content, and the refractive index detector, that's the Optolab, which will be sensitive to both types of materials. It's a universal concentration detector. So between the two of them, we can determine the concentration at each elution volume of protein and of the modifier. Combining that with the light scattering information from the DAWN or mini-DAWN, we can then determine the molecular weight of each of those components. And so here we see across the peak the protein molecular weight, the modifier molecular weight, and the total molecular weight of this species. And here's an example of how this analysis is used to look at the 2 different expression systems for a single glycoprotein. So this protein was expressed in both insect and mammalian cells. And you can see that the 2 types of proteins, or the 2 expression systems, elute at different times in the chromatogram, indicating different molecular weights. But if we do the light scattering analysis, we see that both have identical protein molecular weights, which is as we would expect because they're expressing the same protein. The protein is not oligomerized, it's monomeric in both of these conditions. And they have different degrees of glycosylation, so different amounts of carbohydrate on each of these peaks. In addition to the fact that these two expression systems produce different degrees of glycosylation, we also see the degrees of heterogeneity across each of these peaks. So by looking at the variation in the carbohydrate molecular weight across the peak, each of them we can determine how heterogeneous each of these expression cells produces. So quickly to summarize what we can do with SEC-MALS and SEC-DLS, these provide the absolute molar mass and size of the solution components. They're really important for non-ideal samples and conditions, samples that aren't described well by the standard protein calibration material, which interact differently with the column than the standard globular proteins of different conformations, or chromatography may be not very reproducible, your column may be aging, and you won't notice it unless you actually do a recalibration. These will give us the degree of homogeneity and heterogeneity across the peak, and tell us about aggregated and unfolded species as well as impurities. And then finally, the triple detector system, UV-MALS-RI, will help us analyze the conjugated molecules such as glycoproteins, really important for membrane proteins as well because membrane proteins that are solubilized with a surfactant will elute within a micelle, which is surrounding the protein, and the elution time actually will not be related at all to the molecular weight of the protein. It will be related to the size of that micelle. So this is really essential for anybody working on any of these types of proteins and other biomolecules. If you're interested in more information, I encourage you to go to our website, and this is a really helpful article by Eva Foltestognev at Yale University. She has written up SEC-MALS in Methods in Molecular Biology. Next, I'd like to tell you about CG-MALS, which is the combination of multi-angle light scattering with a composition gradient system, and that's used to analyze interactions. And so this uses the same multi-angle light scattering detectors as SEC-MALS and also uses the UV detector, typically from HPLC system, but replaces the HPLC system, which is feeding the detectors with a separated sample, with the CALYPSO composition gradient system, which uses 3 computer-controlled syringe pumps to mix up to 3 different solutions, typically 1 or 2 different proteins and a diluent, in different ratios, inject into the detectors, flow stops, we wait for equilibration, and acquire the light scattering signal, which is then used in the CALYPSO software to analyze the interactions and determine the appropriate association model and the appropriate KDs and KEs that come out of that. This technique was first pioneered as an automated system by Atri and Minton and is now available commercially from Wyatt Technology. So CG-MALS is a label-free and a mobilization-free technique that measures components in solution. It's used to determine the binding affinity, can also characterize nonspecific interactions, and unique to CG-MALS and one other technique, which is analytical ultracentrifugation sedimentation equilibrium, it can measure the absolute stoichiometry, meaning instead of just determining how many moles of one component bind to how many moles of the other component, which is the ratio, it actually determines how many molecules of each component come together to form a complex, and will analyze their molar mass and size as well. CG-MALS is appropriate for analyzing self-association, hetero-association, even simultaneous self and hetero-association from below a nanomolar and up into the millimolars. And it's appropriate for proteins, peptides, aptamers, all types of biomacromolecules. So here's how we do the analysis of a self-association in CG-MALS. We use the CALYPSO to create a concentration gradient, injecting a series of steps into the detectors and stopping the flow and making the measurements of the light scattering. And in a self-association, the weight average molecular weight of the solution will depend on the type of oligomer that's forming. For instance, is it a dimer or is it a trimer? And on the KD, for instance here 10 micromolars and 1 micromolar. And as the concentration increases, the degree of increase of the molecular weight will change, and the shape of that curve and the height of that curve will depend on the specifics of the molecular weight, the type of oligomer forming, and the KD. Now these molecular weights will translate into light scattering curves that look like this and can be distinguished by the software from each other and analyzed. For hetero-association, we use a method which is known as a crossover gradient. So normally when you think of a hetero-association measurement, you do a titration where you have a fixed concentration of one protein and you titrate in the other protein and you get a sigmoidal curve. Because of the opportunities for creating a larger range of stoichiometric ratios that we get using the CALYPSO system, we can actually probe a different series of compositions which go from completely protein Y, where we measure with light scattering the molecular weight of protein Y, all the way to just protein X, where we measure its molecular weight, and then a series of mixtures where we look for the formation of complexes. And so going to the light scattering that we would expect, depending on the type of interaction, if there's no interaction and these proteins are simply sitting next to each other in solution without doing anything, then we get a linear relationship between the light scattering and the composition gradient. But if complexes do form at a particular stoichiometry, then we see an increase in molecular weight due to those higher molecular weight complexes forming. That increases the light scattering, and the degree of increase of light scattering will tell us about KD. So the higher the affinity, the more complexes form, the higher the molecular weight, and we can analyze that to find KD. The position of that peak will depend on the stoichiometry. So for instance, if we have a 1-to-1, 2-to-1, or 3-to-1 stoichiometry, the position of the peak will shift within that gradient. And you'll notice that the height of the peak of the light scattering will also increase, and that's because the molecular weight of 3-to-1 is higher than 2-to-1 and higher than 1-to-1. If we were to have, for instance, formation of a 2-to-2 complex, it would peak at the same position as the 1-to-1 complex, but it would have twice the light scattering because it has twice the molecular weight. So that's how we can actually distinguish between complexes that have the same ratio but different absolute stoichiometries. So the CALYPSO software will acquire and analyze the light scattering data in order to understand which types of associations are going on, and it will offer all kinds of models that can be applied, for instance in self-association, a monomer-dimer model or a monomer-dimer hexamer model. Basically you can put together arbitrary sets of models based on different types of oligomers. Also an isodesmic, which is a progressive self-association model. In terms of hetero-association, there are basic models like monovalent, AB, or multivalent with equivalent binding sites, so 1 to n with arbitrary n's, and could be 2 or 3 or 4, whatever you like. If these basic models don't work for you, then you can also set up more sophisticated models, which include arbitrary combinations of self and hetero complexes. So you can have simultaneously self-association to form dimers of A, and B might be forming trimers, and A and B might interact with 1-to-1 ratio or some other combinations, and all this can be modeled up simultaneously. There are opportunities for looking at cooperativity and also for multivalent interactions, which are quite complex, and Dr. Hasty will be telling you about some of these a little bit later on. You can also analyze incompetent fractions, so if some of the stock solution is not associating, that's something that can be determined as well using the CALYPSO software. So what do you get in terms of the results? Well, first of all, you get the molecular weights of A and B. And then if you have applied a simple model, for instance, a homodimer or a simple 1-to-n model assuming equivalent binding sites, you'll get the KD that corresponds to each binding site. If you're using a more complex model, you'll get, as a result, a series of association constants, Ka, that correspond to this equation, the mass action equation, with 1 Ka for each complex it forms in equilibrium with the free monomers of A and B. And if you have in this complex model some submodels that include equivalent binding sites, you'll get the Ka for each binding site. We also produce a chi-square value and residuals, so you can compare models and determine the best fit model, and also provide the abundance of each complex at each composition. So this corresponds to the various compositions. For instance, at this particular composition was the concentration of free monomer of A of the 2-to-1 complex, and here of the 1-to-1 complex. And actually you can see that there's very little free monomer of B under this particular condition. But if you go to the central plateau over here, then we can see that we have large amounts of the 1-to-1 and 2-to-1 and small amounts of remaining free monomer. So all this information is provided in the analysis. So to summarize briefly, and there's an entire world there of interactions that you can analyze with CG-MALS. It gives you self-association, so you can analyze the affinity and the oligomeric states of a self-associating system. You can also analyze protein-protein interactions in solution, in free solution, to determine the KDs and the absolute molecular stoichiometries. For complex interactions, this technique gives you intuitive and self-consistent interpretation of the results. And the reason that I say that is because you're getting the molecular weights. So you have the monomer molecular weights, and you know that in order to form a particular complex, the molecular weight of the complex has to add up to the corresponding molecular weights of the number of monomers that go in. And all of this has to be self-consistent. And all of this is provided for you in the software so you can assess the viability of the models and the fit of the results, and is in fact consistent with other information that you have. Again, for more information, I suggest going to the website, and you can also read an article that was published in 2013, Biophysical Review, which is a pretty comprehensive review of this technology. And so, to summarize, here are the answers to these questions. How do we determine the structure and solution properties, the functionality, and the quality of the proteins and biomolecules using light scattering? Here are the techniques, SEC-MALS with DLS as well, and CG-MALS to answer these questions. And with that, I thank you for your attention, and we'll now turn it over to Dr. Hasty.
Our second speaker is Dr. Catherine Hasty of the Scripps Research Institute. Over to Kate.
Great. Thanks, Dan, for that introduction to light scattering in CG-MALS. And I'd like to present our work in the Sapphire Lab here at TSRI that we have done using SEC-MALS and CG-MALS to understand the interaction between the Ebolavirus GP and different therapeutic antibodies. So the first thing I'll talk about is just giving you a bit of background on Ebolavirus GP and tell you how we use SEC-MALS to determine the mass and glycan content of this protein. And thirdly, I'll go through a couple of examples of some CG-MALS experiments that we've done with antibodies against Ebolavirus GP. So Ebola is part of the filovirus family, which includes Sudan, Bundibugyo, and Marburg viruses, amongst others. And it's a significant human pathogen evidenced by the outbreak in 2014, where 30,000 cases arose from a single infected toddler. The Ebola virus looks like this. This is an electron micrograph of the actual virus. And if we zoom in on just one section of this virus and look at this artist rendition of this portion of the virus, we see that there is a single glycoprotein that decorates the virion surface. This protein is termed GP, and this is what actually attaches to and fuses the virus into a new host cell. So GP is a trimeric protein. It's composed of a GP1 subunit, which drives the attachment to the host cell, and the GP2 subunit, which wraps around the base of each of these three GP1 subunits and forms the fusion machinery. Now, on each of the GP1 subunits, there's something called the glycan cap, which is a heavily glycosylated region of GP. And attached to each of these glycan caps is a further glycosylated region called a mucin-like domain. Now this is O and N-linked glycosylation, and they're very, very large. We don't actually know what the mucin-like domain looks like, but from solution scattering experiments, we can see that Ebola virus, as it exists on the on the virion surface, is composed of a proteinaceous core that's surrounded by these heavily glycosylated regions. Now, because GP is the sole protein on the exterior of the virus, it's a significant target for neutralizing antibodies. and how and where these antibodies bind is of major interest, both for therapeutics but also for vaccine design. So we use crystallography and EM here at Scripps for understanding where these antibodies bind. And in fact, we have several structures of GP bound to different antibodies, particularly those that bind to the mucin-like domain, the glycan cap, and those that bind the base of the GP1, GP2 subunit down here called base binders. So the three antibodies I'll be speaking about today are 1H3, KZ52, and 13C6. So to understand how these antibodies bind, we use CG-MALS. There are specific questions that we're trying to answer, including, are these multivalent sites equivalent? So on each GP trimer, there's 3 possible sites for an IgG to bind. And conversely, on each antibody, there's 2 potential sites for a GP to bind. So we want to know, does this actually happen? Do 3 antibodies bind to a GP trimer with equal affinity? And does an antibody bind to 2 GPs at the same time with equal affinity? We'd also like to know how much of each of these different stoichiometric species are there. And lastly, we can also understand what the affinity of each of these species is using CG-MALS. So there's a few pieces of information that are important to know before embarking on a CG-MALS experiment. First, we would like to understand whether or not each component in our experiment is actually homogeneous. This is particularly important for our glycosylated protein. Secondly, we need to know the mass of each component on its own to ensure that the masses that are calculated in the CG-MALS experiment are correct. And thirdly, because of the relationship Dan spoke about earlier, where the intensity of the light scattered is related to the mass of the thing that's scattering the light multiplied by its concentration and refractive index, we need to accurately measure the concentration of the component scattering the light. And to do this, we need to know the true DNDC and the extinction coefficient of the GP. Now, as I mentioned, GP is heavily glycosylated, and so the standard methods one might use for determining mass or the DNDC and the extinction coefficient aren't accurate. And so instead, we use SEC-MALS to determine these components. So our SEC-MALS setup looks like this, where we use a 3-detector system of a UV detector followed by a light scattering and refractometer downstream of our separating column. And for these experiments, we use a modified version of the GP where those heavily glycosylated mucin-like domains are actually removed. So the only glycosylated portion that's remaining intact is the GP2 regions and the glycan cap of GP1. So we can collect our data, and this is what it looks like. So you can see that we can obtain the masses of the glycoprotein, which is 197 kilodaltons, and we can also separate the protein components and the glycan components. And if we look at the protein trimer mass, if we just calculated based on sequence alone, the protein mass obtained through SEC-MALS and what we would expect by sequence agree very well. And we can also extract the DNDC as well as the UV extinction coefficient for this particular protein, which will then be used in the CG-MALS experiments later on. So this is what our CG-MALS instrumentation setup looks like, where the GP and the IgG are mixed with the CALYPSO and sent into the UV and the light scattering detectors, and then the data is collected by the CALYPSO software. Our experimental setup looks like this, where we can perform a homoassociation gradient of either the glycoprotein or the antibody. And we can do this to determine if any of the GP exists as monomers or if it's solely trimeric, or if higher order oligomers beyond trimers are present in our solution. We can also determine if the IgG itself is monomeric or if there's some portion of aggregates. And in between, we have our crossover gradient that combines different amounts of each the glycoprotein and the antibody. Now, as Dan mentioned previously, when the IgG and the GP associate with one another, it produces a light scattering signal that is greater than what we would expect if they weren't interacting at all. So the first data set I'll be speaking about is this antibody called 1H3, which binds to the glycan cap up here on the GP. The first step is to collect our data, and the data we collected look like this. And ideally, each of our compositions would come to a nice plateau, as you see for this first data set, or this first data point. So for this GP antibody complex, the kinetics of association are very slow. And even after 10 minutes of allowing each of these peaks to come to equilibrium, we really don't see a plateau. However, regardless of this, we can still extract useful information from this data. And so our next step is to set the data points for each of these compositions in the gradient. And it's from these set data points that we obtain our experimental data points. So now each of these data points account for a single composition of IgG and GP, and we've plotted it here where the 1H3 concentration is going up and the GP concentration is going down. Okay. Next thing we need to do is create a model to fit the experimental data. There's two components to this section. One is the user input, and here we've created a model that will best represent our data. Our model for this data set incorporates two terms that are described by two equivalent binding sites on each of the IgGs for a GP. And this accounts for both one-to-one associations as well as one-to-two complexes that are in equilibrium with the free GP and the antibody. We also have a component that describes two IgGs bound to a GP trimer that are in equilibrium with free GP and IgG again. So the overall association model for this data set includes one-to-one, one-to-two, and two-to-one complexes. Now once we've input these model parameters, the software then calculates the association constant for each component, which then can be used to calculate the KDs. Now importantly, the molar mass of each component is also calculated and is shown here. And these values should match very well with the expected values for each, which is about 145 kilodaltons for the IgG, which we derived from the sequence of the antibody, and about 197 kilodaltons for the GP, which is what we obtained from SEC-MALS. So if the model is correct, the fit of the curve based on this model should match very well with the experimental data points. And you can see ours does. So from this we can conclude that yes, the multivalent sites are equivalent. Now I will mention that we would expect to see 3 IgGs binding to a GP trimer under these experimental conditions, but this stoichiometry is not populated, likely due to low concentrations of GP in the IgG that we're using. So our model includes all 3 of these components, but I wanted to just demonstrate what the model would look like if we only included one of the components. You can see the green dotted line here what our fit would be if we only included 1 to 2 associations, and in the purple dotted line here what our fit would look like if we only included 1 to 1 associations. So you can see that we really do need all 3 components for our model to best fit our experimental data. So we can also answer the question of how much of each species is there. We can build a model that has all of these different components in it, but are they well represented? And although we do need to include all 3 terms, only a small amount of either the 1 to 2 or 2 to 1 stoichiometries are actually present. And about 20% of it in the bulk in this experiment is the 1 to 1 stoichiometry only. We can also determine what the affinity of each of these associations is. So the antibody can bind to a GP with about an affinity of about 490 nanomoles. And 2 antibodies bind to a GP trimer with an affinity of about 785 nanomoles. So from this data, we learned that 1H3 binds to GP with relatively low affinity, and it does not form any large multivalent complexes. The second data set I'd like to show you is that of KZ52, which binds at the base of Ebolavirus GP and cross-links the GP1 and GP2 subunits together. We're wondering if the affinity is similar to 1H3 or if it's different, and is the binding mode similar or different to that glycan cap binding antibody. So our data looks like this, and you might immediately notice that it looks quite a bit different than the 1H3 data. Number one, we do see very nice plateaus for each different composition, except for this single data set here. Regardless, we can set our data points and obtain our experimental data. And I want to point out that for this peak data set, or this peak data point here, this actually is a very, very large complex. And now the weight average molar mass is plotted against the GP concentration, and our peak is occurring at more than 2,000 kilodaltons. So even though we're using very similar concentrations as we did with the 1H3 experiment, we get very, very different light scattering. Now, we did attempt to create a model to fit this data, and it was actually very challenging, primarily because these proteins are forming such large complexes. What we were able to determine was that we have 1 to 2 complexes, so an antibody combined to 2 GPs at once. We also have a trimer of GP binding to 3 antibodies at once. And in the middle of the light scattering curve, we have this large agglutinated GP antibody complexes where we get our multivalent antibody and our multivalent GP forming these very, very large complexes. From this, we can determine that the multivalent sites are, in fact, equivalent, as we saw with the 1H3 antibody. So how much of each species is there? Unlike the 1H3 data set, where we saw only a scant amount of complexes that were not one-to-one complexes, we see a more normal distribution with the KZ52 antibody, where about 30% of the complexes formed are 1 to 2 associations, and about an equivalent amount, quarter of that, is the 2 antibodies or 3 antibodies binding to a GP trimer. Across the entire gradient, we see 1 to 1 complexes, and the agglutinated GP accounts for about 15%. And it's likely that we see these different amounts of each complex as opposed to the 1H3 data because the KZ52 binds to GP with a much higher affinity of about 15 nanomole. So from this, we concluded that KZ52 combined to GP with very high affinity, and it agglutinates the GP to make these very large complexes. And the distribution of species is normal. So the last data set I'd like to show you is that of 13C6, which, like 1H3, binds to glycan cap of GP. So these antibodies have an overlapping epitope. They both bind right at the top of the GP trimer. So we were wondering, is the binding mode and the affinity same or different? So here's the data that we collected. And again, we see nice plateaus for this particular antibody. We set our data points and obtain our experimental data. And because it was a similar binding location as 1H3, the first model that we attempted to use to fit this data is that of the just simple 1 to 2 and 3 to 1 associations. However, this fit was very poor to our experimental data. So in fact, it seems that the multivalent sites are not actually equivalent. Instead, our model needed to include 1 to 1, 2 to 1, and 3 to 1, 3 to 2 complexes of each of these species. And we looked at how much of each of these species there was. We saw something really interesting. So the vast majority of the species we saw were the 1 to 1 associations. About 10% of the species were 2 antibodies binding to a GP trimer, and only a very, very small amount accounted for 3 antibodies binding to 2 GP trimers. So this already gave us an inkling that something very interesting was going on. Notably, there was a complete lack of any antibody bound simultaneously to 2 GP trimers. When we looked at the affinity of each of these species, we noticed that the 1-to-1 stoichiometry had an extremely high affinity of sub-nanomolar. However, the other 2 fit models we had for this particular data set was much higher. So 2 antibodies bound to a single GP trimer with an affinity of about 300 nanomolar, and the 3 to 2 association had an affinity of about 14 nanomolar. So this large difference in affinities, combined with the absence of the 1 IgG binding to 2 GP trimers, made us question whether or not our hypothesis for these binding geometries was actually correct. Did we need a new hypothesis that maybe accounted for this antibody binding to the GP bivalently? To answer this question, we turned to EM, where we took the entire IgG and bound it to a GP trimer and saw that, in fact, yes, each arm of this particular antibody simultaneously bound to the same GP trimer. So instead of having a single arm engaged in the GP trimer, this antibody engages two of the GPs at the same time. So now a revised model includes this very high affinity bivalently bound antibody plus a bivalent bound antibody to the trimer that has another antibody coming on with much lower affinity. And then this is now what we think is an average affinity. So you have two bivalently bound antibodies to a trimer, which are the very high affinity associations that are then cross-linked by a much lower affinity interaction with the third antibody coming on. Now we also analyzed this GP13C6 interaction using another solution-based technique called ITC. And it's worth pointing out that the average stoichiometric ratio of 1.2 corresponds to the CG-MALS result where we have mostly 1-to-1, but also some 2-to-1 and 3-to-2 associations as well. However, ITC does not have the capability for identifying and extracting the KDs of these multiple complexes that are occurring simultaneously in the solution. So the KD for the 13C6 GP interaction obtained through this technique is quite a bit higher than that we obtained for the CG-MALS experiment. So what did we learn from CG-MALS? For the KZ52 antibody, we learned that it binds to GP with high affinity, it very efficiently cross-links the protein, and that the multivalent sites on each protein are equivalent. We also saw that we have, for these two antibodies that bind in the same location on the GP, they do so in very different ways with very different affinities. So for 1H3, the multivalent sites are equivalent. However, they bind to the GP with quite poor affinity. However, the distribution of species is normal. For 13C6, we got a surprise and found out that it was a bivalently binding antibody, and that 1-to-1 associations are extremely strong and account for the bulk of the interactions. We do see other stoichiometries, but they have much lower affinity and are poorly populated. So our lab uses light scattering for multiple reasons. First, we use SEC-MALS to characterize Ebolavirus and other viral glycoproteins. We do this to understand their homogeneity, their oligomeric state, as well as the glycan content. We use CG-MALS and EM synergistically so that we can characterize these complex interactions between glycoproteins and therapeutic antibodies. And importantly, another solution-based technique that we use in the lab didn't provide the key information for deciphering these particular interactions. Lastly, understanding the fine details of how an antibody interacts with glycoproteins is a crucial part in characterizing these therapeutic candidates. So with that, I'd just like to thank the folks who were involved in this work, particularly those in the Ullman Sapphire Lab and the Ward Lab who did the structural work, as well as those who identified the antibodies and produced the antibodies for us to use, and as well as the folks at YI Technologies for their great customer support.